In this video at Medical Sciences, we are going to take you through digestion, absorption, and metabolism of lipids in the body. So as we begin, we urge you to subscribe, like the video, and share to many others. Want to see when we take food which contains lipids like triglycerides, like cholesterol, and phospholipids. So in other words, the food which we take contains triacylglycerol, contains cholesterol, and contains phospholipids. So these are the major components we eat in food when we take a fatty meal. So we want to see how these food products which we take are digested, absorbed, and utilized in the body. So we're going to begin in the mouth, because digestion begins in the mouth, whereby in the mouth we have physical digestion, which is due to the action of teeth, and we call it mastication. Mastication occurs due to the action of teeth and the cheeks. Then after breaking down these big food particles into simpler particles by the action of teeth is what we are calling mastication, or you can call it chewing. Then after chewing, the food is rolled into a food bolus by the tongue. This is the action of the tongue. And this food bolus, which is formed, so if this is the food bolus, which is formed in the mouth, it is swallowed via the esophagus, and this swallowing to the stomach is by peristalsis. So the food is then swallowed by peristalsis. But before swallowing, there is also chemical digestion, whereby chemical digestion, it is dealing with that in the saliva, there is an enzyme called salivary, or what we can call lingual lipase. There is an enzyme called lingual lipase in the saliva, but this lingual lipase acts best in acidic media. And so the action of lingual lipase is going to break down short chain tags are broken down by lingual lipase to MAG plus free fatty acids. This MAG is known as monoacylglycerol. So the lingual lipase, which acts in the stomach, so meaning when I'm chewing food, I mix it with the saliva. After swallowing by peristalsis, it is activated in the stomach due to presence of HCl. So meaning the lingual lipase comes together with food and is able to work on the short chain. The pregnant point here is it works only on the short chain tags, or triacylglycerol to monoacylglycerol and free fatty acids. Then when the food reaches in the stomach, in the stomach, the presence of food is going to trigger. So when the food reaches in the stomach, it triggers the production of gastrin hormone, and this gastrin hormone triggers production, which triggers production of gastric juice. And this gastric juice contains substances like HCl, that is hydrochloric acid, and it contains gastric lipase. It contains an enzyme known as gastric lipase, whose action is like lipase. So we're going to see the hydrochloric acid which is produced is going to provide acidic media, provides acidic pH, and then this acidic pH favors both lipase and gastric lipase. So we are seeing also gastric lipase working on the short chain, short tags, breaking them down into monoacyl glycerol plus free fatty acids. And the enzyme is gastric lipase. So, and this reaction is a hydrolysis reaction. This one still occurs in presence of water, and this one also occurs in presence of water, because they break the ester bond by adding water, but working on the short chain triglycerides. So in the stomach, we see majorly physical. Physical is the churning action, whereas chemical is the action of hydrochloric acid, which provides acidic media, and then gastric lipase, which works on the short chain tags to monoacyl and free fatty acids. From there, this food chain continues to the duodenum. And within the duodenum, duodenum is the first part of the small intestine. So, in the duodenum, the acidic food chain, this is food acidic, because the food from the stomach, after being acted on, it comes containing the acid. So when it reaches within the duodenum, the acidic food chain is going to trigger production of hormones like CCK and secretin. These are the hormones produced, cholecystokinin and secretin hormone. And once produced, the CCK and secretin, they move to the pancreas. CCK and secretin go to the pancreas and the gallbladder. In the gallbladder, it is going to trigger production of bile salts. So it triggers production of bile salts and also triggers production of pancreatic juice. That is the role of these hormones, CCK and secretin. They go to the gallbladder, triggering production of bile containing bile salts, and also they go to the pancreas, triggering production of pancreatic juice. And this bile salt, its role is enabling emulsification. It is responsible for emulsification of fats. How does it do it? Emulsification means the fats which you have eaten are very big in size. So the bile salts break this big fat globule into small fat droplets called micelles. These small lipid droplets, they increase the surface area for the action of pancreatic enzymes from the pancreatic juice. And this pancreatic juice contain components like the bicarbonates. These ones, they provide alkaline media by neutralizing the HCl from the stomach. They contain enzyme like pancreatic lipase. They contain cholesterol esterase enzyme. They also contain an enzyme known as phospholipase A2. So these are the hydrolytic enzymes found in the pancreatic juice, which is produced by the acinar cells, triggered by secretin and cholecystokinin. And these enzymes, are assisted by the bicarbonates and biosalts, where the biosalts breaks down big fat globule into smaller droplets, whereas the bicarbonate provides the alkaline pH for the action of these enzymes. And we're going to see that pancreatic lipase is going to break down triglycerides. So the triglycerides, both the short chain and the long chain, are finalized and broken down to monoacylglycerol plus 2 free fatty acids. And the enzyme is pancreatic lipase in the presence of water. So the reaction is hydrolysis. So it catalyzes the hydrolysis of long chain and short chain tags to monoacylglycerol and to free fatty acids. Whereas cholesterol esterase catalyzes the hydrolysis of cholesterol esters and converts it into free cholesterol plus free fatty acids. Because we said the food we take contain triglycerides, contain cholesterol, and contain phospholipids. So the cholesterol is acted upon by cholesterol esterase, or you can call it hydrolase. It acts in the presence of water, so it is still a hydrolysis reaction whereby it breaks the ester bonds, forming free cholesterol and free fatty acid. Whereas the phospholipase acts on the phospholipids, because the same food we take contain phospholipids, and these phospholipids are broken down by phospholipase A2 to lysophospholipid plus free fatty acid. And the enzyme that is catalyzing this reaction is phospholipase A2 in the presence of water. And this one will mark the end of digestion. So under digestion, we have seen in the mouth, apart from chewing and rolling of the tongue, we have lingolipase acting on the short chain tags in the stomach. In the stomach, we have seen also there is production of gastric juice, which contains HCl that provides acidic media, together with gastric lipase that breaks down short chain tags to monoacylglycerol and free fatty acids. Then the arrival of the acidic food chain in the stomach, in the duodenum, which is the smaller part of the first part of the small intestines, triggers production of the duodenal cells containing CCK and secretin, which are the hormones responsible for triggering production of bile salts from the gallbladder and pancreatic juice. Bile salts breaking down lipid droplets to micelles, increasing the surface area, the bicarbonates providing alkaline pH, whereas the enzyme phospholipase, pancreatic lipase, hydrolyzing tags to MAG and to free fatty acids, whereas cholesterol esterase catalyzes the hydrolysis of cholesterol esters to free cholesterol and free fatty acids, whereas phospholipids are hydrolyzed by phospholipase A2 to lysophospholipid and free fatty acids. So this one marks the end of digestion of lipids. From there, we shall look at absorption. How are these products, the fatty acids, the monoacylglycerol, the free cholesterol, and the lysophospholipid, how are they absorbed in the body? So we are looking at absorption. And under absorption of lipids, we are saying if this is the lumen, and then this one is going to be the enterocyte or the intestinal mucosa. So this is the intestinal mucosa cells. This is going to be the lumen, and this is going to be the apicocyte. So this is the intestinal lumen, where we are seeing absorption taking place in the ileum, takes place in the ileum, whereby in the rumen, so the final products, we have the short chain fatty acids, free fatty acids. The short chain means those ones less than 12 carbon atoms, together with the glycerol which might come from there, the glycerol which we might eat in the food. These ones are absorbed directly. These ones are absorbed directly into bloodstream and taken to the liver via hepatic portal vein. That is point number one to note under digestion. That short chain fatty acids, which have carbon atom 12 and the glycerol, for them they are absorbed directly to bloodstream by diffusion and taken to the liver via hepatic portal vein. Whereas the long chain fatty acids plus MAG together with lysophospholipid plus cholesterol esters, these ones they combine with vitamin A, vitamin D, E and K. They combine with vitamin A, D, to form what we call mixed micelles. So these mixed micelles containing long chain fatty acids, containing monoacyl glycerol, containing lysophospholipid and free cholesterol, they cross the enterocyte. This is the intestinal wall by diffusion, and when they reach, we are going to see the long chain free fatty acids combine together with MAG, and they are reconverted to form tags. So we are seeing reconversion. Here the point is reconversion. The free fatty acids combine together with MAG to form triglycerides, whereas the lysophospholipid, which was formed, is converted back to phospholipid. The cholesterol, the free cholesterol, which was formed, is also reconverted to cholesterol ester. So the long chain, the MAG combined to form tags again. The lysophospholipid converted back to phospholipid. So the free cholesterol converted back to cholesterol esters. So we are seeing the law of absorption in the duodenum was to enable absorption. After absorption into the enterocytes, they are reconverted. So we are seeing at the end, we have the tags, we have the phospholipids, we have the cholesterol esters, but 90% is triglycerides. So these triglycerides which are formed, they combine with a lipoprotein, and this lipoprotein is known as the chylomicron. They combine with the chylomicron, and this chylomicron has an apoprotein known as ApoB48. So this lipoprotein, after forming tags, phospholipids, and cholesterol esters, they are transported via the lacteal. You remember the villus has the lacteal. Then it moves through the lymphatic vessels, then to the thoracic ducts. These thoracic ducts are the blood vessels near the heart. Why is it not taken directly into bloodstream? This chylomicron is very big, so it cannot maneuver into the capillaries within the microvilli. So what happens is that the lacteal is very big, so they pass through the lacteal to the lymphatic vessels, then to the vessels near the heart, where they are able now to enter the blood circulation, where the capillaries are very big. So that is the transportation. After being absorbed, the tags, the phospholipid, the cholesterol ester, combined with the chylomicron lipoprotein, having ApoB48, and are transported via the lacteal to the lymphatic vessels, then to the thoracic ducts, which are the vessels near the heart, and then they are able to enter general saturation. So from the thoracic ducts, the chylomicron, which are combining with the tags, phospholipid, and the cholesterol ester, enter blood saturation from the thoracic ducts, which are the vessels near the heart. So after reaching there, we are going to see the chylomicron as it is moving into blood saturation. So as the chylomicron, after reaching general saturation, it has moved. We have got the chylomicron moving via the lacteal to the lymphatics, to the lymphatic vessels, then to the thoracic, and then finally into the general saturation. And when it reaches the general saturation, it is going to be distributed. The components of the chylomicron, one, it moves to the skeletal muscle. If there is exercise and there is need of energy, it is also taken to the adipose if non-energy is needed for storage. Then the chylomicron remnant is taken to the liver. So to the liver we see the remnant, the chylomicron remnant, the one that is transported to the liver. And as the chylomicron is moving in the saturation, it is the one that is responsible for making blood. When you collect a blood sample after 2 hours of eating a fatty meal, blood will appear milky due to the saturating chylomicron containing triglyceride 90%, cholesterol 5%, and phospholipid 7%. So it is the one that makes blood to appear milky. So we are seeing chylomicron after reaching general saturation, one destination is taken to the adipose for storage, or taken to the skeletal muscle to provide energy in case of exercise. Then the remaining balance is transported to the liver, and it is the one that the chylomicron makes blood to appear milky 2 hours after a fatty meal. So that is the destination of the chylomicron containing apoprotein B48. So this one ends what we call absorption. Absorption is that short chain and glycerol are absorbed directly and taken to the liver by simple diffusion. The long chain fatty acids, the lysol, and the free cholesterol combine with the vitamin A, D, E, and K to form mixed micelles, which are absorbed into the enterocytes by diffusion. And within the enterocytes, they are reconverted back, whereby these ones form tags, lysophospholipid to phospholipids, then cholesterol to cholesterol esters. Then they combine with the protein known as the chylomicron, which is very big in size that it cannot enter directly into bloodstream. So it moves via the lacteal to the lymphatic vessel, then to the thoracic ducts, which are the vessels near the heart, and distributed to the adipose, skeletal muscle, and the remaining balance is taken to the liver. And when it reaches in their destination, we are going to see how are lipids utilized. So the last part is utilization. Utilization. or what we can call metabolism. You can call it utilization, you can call it metabolism, or you can call it fat. And the utilization of lipids in the body is both catabolic and anabolic reactions. And it is triggered by starvation. Whenever there is starvation or increased exercise, someone has been doing strenuous exercise, these two, they trigger production of hormones. They stimulate production of hormones like epinephrine, hormone like glucagon, hormone like thyroxine. So these hormones, when they work together, they go to the adipose because the site of lipolysis is the adipose. When they reach in the adipose, they stimulate a hormone known as hormone sensitive. They stimulate hormone sensitive lipase. In the adipose, there is an enzyme known as hormone sensitive lipase. Without these hormones, it is inactive. But once there is starvation and increased exercise, these hormones are produced, which go and activate hormone sensitive lipase. But there is also another form of lipase in the blood vessel known as lipoprotein lipase. So these two hormones are responsible for lipolysis. They are the one that do the work of lipolysis or lipolysis, whereby that there is need of energy. The triglycerides in the adipose tissue, they are converted or they are broken down to free fatty acids, 3 of them, plus glycerol. And the enzyme that is catalyzing this reaction is lipases in the presence of water. This lipase can be hormone sensitive lipase, it can be lipoprotein lipase. So when they break down triglycerides to free fatty acids and glycerol, this process is what we are regarding as lipolysis, because we need energy when there is starvation, we need energy when we are doing exercise. So after getting these products, that is free fatty acid and glycerol, the free fatty acid, which is produced as a product, is transported bound to albumin and taken to the liver and skeletal muscles. And within the mitochondria of the liver and the skeletal muscles, this free fatty acid undergoes beta oxidation. It undergoes beta oxidation to form what we call acetyl coenzyme A. That is the fate of free fatty acids. That free fatty acids, after being produced, transported bound to albumin, taken to the skeletal muscles and the liver mitochondria, where they undergo beta oxidation to produce a component known as acetyl coenzyme A. So the acetyl coenzyme A produced, this acetyl coenzyme A is responsible for many metabolic processes. One of them is that we can, in case we need energy, it can go to Krebs cycle. And in a Krebs cycle, it is able to provide energy in the form of ATP, water, and carbon dioxide. So when there is a need of energy, the acetyl coenzyme A produced from free fatty acids goes to the Krebs cycle to provide energy. But in case there are cells in the body like brain and red cells that lack mitochondria and they cannot utilize this energy. So what they do, this acetyl coenzyme A can undergo ketogenesis to form what we call ketone bodies. Ketone bodies like acetone, acetoacetate, and beta hydroxybutyrate, which can be broken down by the process known as ketolysis to provide energy to the brain and red cells that lack mitochondria. And that's why sometimes when you find someone has been sick, not eating, you do their urine analysis, you find the ketones present. And even during pregnancy, they always have ketones showing there is lack of energy. Then, if there is no need of energy, this acetyl-CoA can still undergo lipogenesis, whereby we can use it to produce other tags for storage. If there is no need of energy, it undergoes lipogenesis. Then if maybe the body needs cholesterol to form steroid hormones like testosterone, estrogen, maybe we need bile salts, bile acids, or we need fat-soluble vitamins, it undergoes a process known as cholesterol synthesis, whereby this cholesterol formed can be utilized for formation of hormones, steroid hormones like testosterone, vitamin A, and bile acids. And even it can form part of the cell membrane. That is the fate of cholesterol which is synthesized. Then another thing we can see is that this acetyl-CoA can undergo also gluconeogenesis. It can undergo gluconeogenesis to produce glucose, which we can use for storage. And lastly, we can also utilize it for formation of amino acids, which are part of protein. So this is the fate of free fatty acid. And I want you to note that a single free fatty acid can produce up to 131 ATPs when taken to Krebs cycle and electron transport chain. So the processes here is acetyl-CoA enzyme A, which is a central point for all metabolic processes, can be used to provide energy. Ketone bodies can be used to get fats by lipogenesis, synthesize cholesterol, synthesize amino acids, and also get glucose by gluconeogenesis. And lastly is the fate of glycerol. So the glycerol which is formed for it, because we have looked at the fate of the free fatty acid which was formed from lipolysis. Then what about the glycerol? So for the glycerol, is taken to the liver, whereby in the liver it undergoes gluconeogenesis. It undergoes gluconeogenesis to produce glucose, because gluconeogenesis occurs only in the liver because of the presence of glucose 6-phosphatase, which is only found in the liver. And how does it that when we have glycerol, it is converted to glycerol 3-phosphate by an enzyme known as glycerol kinase, whereby it utilizes ATP, whereby we add ATP to form ADP. So ATP donates 1-phosphate at carbon number 3 of glycerol to form glycerol 3-phosphate in the presence of glycerol kinase enzyme. And this glycerol 3-phosphate formed can be converted to dihydroxyacetone. Dihydroxyacetone, which is an intermediate, or we can utilize it by converting it to glycerolaldehyde 3-phosphate, which is an intermediate in glycolysis. So that is how glycerol undergoes gluconeogenesis. How do we do this? Glycerol 3-phosphate undergoes oxidation, whereby we utilize NAD+ to remove hydrogen to form NADH in the presence of glycerol 3-phosphate dehydrogenase. This one in food is known as dehydrogenase. It catalyzes the oxidation of glycerol 3-phosphate to dihydroxyacetone. And this dihydroxyacetone, which is an intermediate in Krebs in glycolysis, can even be interconverted to glycerol 3-phosphate. And this glycerol 3-phosphate can be used to produce glucose in the liver by gluconeogenesis, which we talked about here. Or we can use it directly to produce ATP, whereby we can convert it to pyruvate when there is immediate need of energy. So this one marks the end of utilization of lipids in the body. A simple recap is that when there is starvation or exercise, these hormones, epinephrine, glucagon, are produced, which activate hormone-sensitive lipids, and utilization occurs majorly in the adipose tissue, whereby triglyceride is hydrolyzed to glycerol and free fatty acid by lipases, either hormone-sensitive lipase or lipoprotein lipase. Then the free fatty acid is taken to the liver and the skeletal muscles and undergo beta oxidation. This beta oxidation produces acetyl coenzyme A, which is used to provide energy when there is need via Krebs cycle. It can undergo ketogenesis to form ketone bodies, which can undergo ketolysis to provide energy to the brain, which lack the mitochondria. And if there is no need of energy, converted back to the tags and stored. And if we need cholesterol for hormones like testosterone, fat-soluble vitamins, and bile acids, we form it. Then also we can use it for producing amino acids, together also glucose by gluconeogenesis. And the phospholipids for them, they form part of the cell membrane, whereas the glycerol undergoes gluconeogenesis in the liver, whereby it first forms glycerol 3-phosphate and then oxidizes to dihydroxyacetone, which is converted to glycerol 3-phosphate and can be used to produce glucose by gluconeogenesis, or directly produce energy by being converted to pyruvate. Then it can be able to produce energy. Thank you so much for learning with us, where we have looked at both digestion, absorption, and utilization of lipids in the body. Hope you guys have enjoyed it and you have learned something from us. Always keep tuned, subscribe, turn on your notification, and share to many others that we may continue to learn with us at Medical Sciences. Always proud of you, and may the good Lord bless you.